Hey everyone, and welcome to this explainer. I am super excited to have you here today because we're about to embark on a truly fascinating microscopic journey. We're going to strip back the complex layers of biology and look at the very foundation of how life actually works. We'll be moving from invisible microscopic structures all the way up to groundbreaking modern medical treatments that could, quite literally, save your life one day. So let's just dive right in.
Think about this for a second. What is this single building block that makes up every living thing on Earth? I mean, whether it's a massive, towering oak tree, some microscopic bacteria, or you and me sitting here right now, we all share this exact same foundational starting point. And the answer, of course, is the cell. Here's our roadmap for today. We'll start with the building blocks of life, then compare animal versus plant cells, explore the magic of differentiation, figure out what are stem cells, and finally look at stem cells in medicine. We're building from basic biological factories up to some absolute medical marvels.
All right, part one, the building blocks of life.
To truly grasp how biology works, try picturing a typical animal cell not just as some random microscopic blob, but as a bustling, highly organized, tiny factory. Seriously, it's like a tiny city in there. Every factory needs a security gate, right? Well, that's your cell membrane. It tightly controls exactly what substances are allowed to enter and leave. Inside, filling up the space, you have the cytoplasm. Think of this jelly-like mix of water and dissolved solutes as the factory floor itself. This is where the heavy lifting happens, the site of tons of crucial chemical reactions.
To keep this whole factory analogy going, let's look at the specialized machinery on that floor, which we call organelles. The nucleus, that's the boss, the command center. It holds all your genetic material, the literal blueprints for the cell. Then you've got the mitochondria. These are the power plants of our factory, acting as the site for aerobic respiration to generate the energy needed to keep things running. And finally, the ribosomes. You can picture these as the assembly line because this is the specific site of protein synthesis. Every single part has a highly specific, totally vital job.
Which brings us to part two, animal vs. plant cells. Now you and I can walk around to find food and shelter when we need to. A plant obviously cannot. Because of a plant's stationary, sun-absorbing lifestyle, its factory layout requires three major structural upgrades compared to an animal cell. First up, chloroplasts. These contain chlorophyll to absorb light for photosynthesis, literally making food directly from the sun. Second, plants need to stand tall, but they don't have a skeleton, so they have a rigid cell wall made of cellulose surrounding the membrane. This gives the cell its shape and stops it from bursting. And third, they have a permanent vacuole filled with cell sap. This basically pushes the cytoplasm against the cell wall to keep the whole cell nice and firm. So while both animal and plant cells share the same basic core structures, the nucleus, cytoplasm, membrane, mitochondria, and ribosomes, the plant cell has these brilliant adaptations like the cell wall and chloroplasts that animals simply don't need. And sure, an animal cell might have small, temporary vacuoles here and there, but the plant relies on a large, permanent one for its actual structural support. Just a quick aside before we move on. How do we actually study these microscopic factories? Well, viewed under a standard light microscope, we see the broad strokes. But some crucial parts of the machinery, like ribosomes or the inner workings of mitochondria, are simply way too small to be seen without stepping up our game. We have to rely on the massive magnification of electron microscopes to truly reveal this hidden level of biological tech. It's pretty amazing what they can show us. Okay, part three, the magic of differentiation. Up to this point, we've basically looked at generic, standard cell layouts. But an entire complex organism isn't just made of identical basic factories. They have to adapt and change over time to perform highly specific jobs. Cell differentiation is the process where a cell changes to develop the exact structure and characteristics it needs to carry out a particular function. When a cell differentiates, it physically alters its composition. And here's a really cool difference between our two biological kingdoms. In animals, most of this differentiation happens very early on in development, and the cells lose this ability as the animal ages. Plants, on the other hand, retain the ability to fully differentiate throughout their entire lives.
Let's look at how this plays out in real specialized cells, because the structural tweaks are just wild. Ciliated cells literally grow hair-like extensions to sweep mucus away from our lungs. Nerve cells become massively elongated just to conduct electrical impulses across the entire body. Red blood cells, and this one is my favorite, they actually ditch their nucleus entirely, just eject it so they can pack in more oxygen-carrying hemoglobin.
And in the plant world, root hair cells grow these long extensions to absorb maximum water, while palisade cells pack themselves completely full of chloroplasts to catch every single ray of sunlight. Basically, structure dictates function.
Moving right along to part 4, what are stem cells? So if differentiation is a cell locking in its career path, what do you call a cell that hasn't picked a job yet? That's a stem cell. By definition, it's an undifferentiated cell capable of dividing by mitosis an unlimited number of times. These things are entirely unspecialized blank slates. When a stem cell divides, it can either make more identical, unspecialized stem cells, or it can differentiate into a specialized cell, like a nerve or a muscle cell. But you should know that not all stem cells are created equal. Embryonic stem cells, which are sourced from embryos, possess immense, literally unlimited potential. They can differentiate into absolutely any cell type to produce all the tissues in a developing organism. Adult stem cells, though, which we find in tissues like bone marrow, have restricted capabilities. They can only differentiate into a limited number of specialized cell types, and they're mostly just used for replacing and repairing damage in the body.
Which brings us to our final section, stem cells in medicine. Understanding all this microscopic machinery isn't just textbook trivia. It's the foundation for some incredibly active therapies. Stem cell therapy is literally saving lives right now by replacing broken or failing cells. For example, doctors can take adult stem cells from bone marrow and use them to produce fresh red blood cells for patients suffering from anemia. Or they can generate new white blood cells to boost the immunity of someone who's really vulnerable. They can even create platelets to treat severe blood clotting issues. It's quite literally biological repair at the most fundamental level.
Of course, it's not quite as simple as just swapping out spare parts. The therapy faces massive biological hurdles. When you use donor stem cells, the patient's immune system might look at the new cell's different surface proteins, their antigens, and see them as foreign invaders. That leads to dangerous cell rejection, and the patient has to go on heavy immunosuppressant drugs. That is exactly why using a patient's own adult stem cells is vastly superior whenever it's possible. Because those cells share the exact same genes and antigens, there's zero risk of immune rejection. But remember what we said earlier, adult stem cells are limited in what they can become. Embryonic stem cells can become any cell, which makes their medical advantage absolutely unparalleled when it comes to treating complex diseases. However, this creates a really profound societal dilemma. On one hand, the medical potential is staggering. But on the other hand, obtaining these cells requires growing embryos in a lab, which crosses major ethical boundaries for many people who object to the destruction of potential human life. It is an incredibly sensitive, deeply debated topic at the very edge of modern science.
And that brings us to the end of our explainer. I want to leave you with this thought. As medical science advances, how will we balance the incredible life-saving potential of stem cell therapy with the complex ethical boundaries of human life? It's one of those big questions where biology, medicine, and our deepest human values all collide, and it's a conversation society is going to be grappling with for a long, long time. Thank you so much for joining me on this deep dive into the blueprint of life. Keep questioning, keep learning, and I'll catch you in the next explainer.